Beautiful, isn't it? For some, this may just look like a complex string of random bars. However, this is arguably the most important molecule that has ever existed and is responsible for both you and me. While this molecule is important, it is certainly not simple. In fact, the actual structure of DNA was not known until about 60 years ago. This molecule is in every single cell in your body, and if we untangled and stretched each string out from every single cell in your body, it would reach to the sun 600 times. And yes, this molecule is DNA.
It might seem crazy what I'm about to say. Sunshine, she's here.
Hello and welcome to BioChio. Today we will be discovering the structure of DNA with our friends James Watson, Francis Crick, Rosalind Franklin and Maurice Wilkins. We will look at the importance of their work in determining the structure of DNA and the impact of the quality of collaboration and communication on their scientific research. However, to do this, we're going to go back about 60 years to a time where the structure of the most important molecule in life was still unknown. Oh, does my hair look alright? Oh hey look, it's my good friends Watson and Crick! So Crick, you and Watson have been working together since the 1950s and studied at the Cambridge University. What have you contributed to the structure of DNA?
Contributed? We practically designed this baby.
Well, first, a little bit of history. In 1943, Oswald Avery proved that DNA carries genetic information. Most people at the time thought that this would be a protein, not a nucleic acid. By the late 1940s, DNA was accepted as a genetic molecule. Scientists still needed to figure out this molecule structure to be sure. And understand how it worked? That's where we came in. Most people didn't know if this helix would be double, triple, or single.
Yeah, so boom, we come in and join the race to discover the structure of DNA against Rosie and Wilkins. So in 1951, I attended a lecture of Rosie's, and she had found that there's two forms of DNA, and they alter depending on the surrounding humidity of the air. So this helped to deduce that the phosphate part of the molecule is actually on the outside. So I returned to Crick to create a new model of DNA based on this. But, well, my memory was a bit muddy because, I mean, she put no effort to do a clearance. Like, she could be so pretty if she tried. So, yeah, and then we made the model, it turned out to be a fail.
Yeah, so one day in January 1953, Wilkins was really frustrated with Rosalind, and good old Watson here got Wilkins to show us Rosalind's X-ray photos of DNA. This sparked a light bulb moment.
So you see here, the cross over the middle picture basically spells out helix for scientists. So based on this, we designed a new model, the model that was finally accepted as it fit all experimental data.
So there are two chains of nucleotides, one going up and one going down. The base pairs interlock in the middle to keep the distance between constant. Adenine and thymine match together, and cytosine and guanine match too.
Oh, I see. Well, it looks like you guys had a very important role in determining the structure and function of DNA. With that said, I'm going to go see my great friend Rosalind Franklin.
Hi, I can't talk much at the moment. I'm studying viruses and their infectious element.
At what age did you know you wanted to become a scientist?
At the age of 15, I'm very passionate about what I do. I specialise in X-ray crystallography, especially finding the structure of DNA. So I took two photos of X-ray crystallography of DNA, Form A and Form B. While I studied my focus on Form A, it turned out that the longer, thinner line of DNA, Form B, was the one I should have been studying on.
That's a big shame, seeing as you have put lots of effort and dedication into this.
Yes, I know.
Thanks for your time. Would you happen to know where your partner Wilkins is?
We didn't collaborate very well or communicate. I bet he's busy working on something. You should come back in a few years.
Wilkins, I see you have a Nobel Prize.
Yes, I was awarded this alongside Watson and Crick for discovering the structure of DNA. And I made the first accurate model in 1953 and checked it against the diffraction data in Franklin's Photo 51. This was a great breakthrough in science. So, yeah, oh gosh. So this is Watson, Crick and I, and a few randoms. And yeah, we did a great job.
Didn't you collaborate with Franklin though?
Well, she died.
Oh really, from what?
Oh, I don't know, ovarian cancer or something, from working with all those x-rays. I told her to wear that safety gear. I mean, I asked her to collaborate in 1950, but she rejected me. We had very different personalities. She was always direct, honest, and unfair of argument. But between you and me, I reckon that if we got along and collaborated earlier, we would have beaten Watson Crick to the chase.
Whoa, I'm back! discovery has been called the most important biological work of the last 100 years, and the field it opened may be the scientific frontier for the next 100. By 1962, when Watson, Crick and Wilkins won the Nobel Prize for Physiology and Medicine, Franklin had died. All four scientists had an important role in determining the structure of DNA. However, if they had all collaborated and communicated together, they would have found the structure of DNA much more earlier. However, if Rosalind Franklin were alive, would she have been included in the prize?